It's a fantastic honor to be able to give this lecture. I give my very first lecture as a second-year PhD student at Society of Microbiology, General Microbiology meeting at Imperial College, and I never thought that many, many years later I'd be doing this lecture, and particularly Marjory Stephenson, who actually she was elected in 1945, and it's remarkable that took that long for somebody like that, a woman, to get into the Royal Society. My claim to fame is that I'm the only person from Scunthorpe to get into the Royal Society. It's not quite the same accolade, but there may be reasons behind that as well, but there we are. So what I thought I'd do is I'm talking about genomics, but I'm really going to be talking about one area, but this is sort of a snapshot summary of my career, really. I was very lucky that when I walked in for the first day of my PhD, they told me my supervisor had gone on sabbatical and I was no longer going to work with him. And I thought, is that normal? But it turned out, actually, that the person who was waiting to take me on was this guy, Dave Sherratt, who was absolutely just come across back from the US, and one of the most brilliant people I've ever met, and turned out to be a fantastic PhD student, really looking at the molecular basis of how DNA recombines. Very pure, basic DNA type PhD, and it set me off on a great track, really. And then I was very fortunate. I met this guy, Stanley Falco, before he was very famous, and I intuitively knew that he was the person who I wanted to work with, and went over to the University of Washington and worked with Stanley for a while. And I sort of went this surgeon into Ireland, so welcome back to Ireland. I don't know if many of you know that I did work as a lecturer. I was a lecturer at 26 years of age, which I think you probably can't do nowadays, but I spent 5 years here at the Moyne Institute, and they're having their 100th anniversary, and that was John Arbuthnot who looked at it. But then I went off into industry. I went for 10 years in industry. And that sort of shaped my career, really, because I learned how to make medicines, and I learned really about that, how to apply science and technology in the field, and it sort of shaped my career ever since then, even though I did return eventually back into, I went back to Imperial, and this is a building, actually, which I raised the money for. I raised £20 million, and the interesting thing is the university, you think, well, if you raise all this money for a building, the university is going to throw gallons of flowers over you and everything, but the university totally ignored me, and just, actually what happened was they put there's a receptionist in the building when you walk into any building, and 3 months into my moving into the building, the university sent me a bill for the receptionist to charge to my grants. Then I realized, well, universities are quite different from what you might expect. But the message there is, don't always do things because you expect to get the accolades. Sometimes you don't, but they're worth doing anyway, because the building's still there. Then I went off to the Sanger, and I spent 14 fantastic years, very privileged years at the Sanger, and now I'm back in the University of Cambridge. So that's the summary of my career, if you like. And what I'm going to do is I'm going to talk about genomics, but I'm going to focus on one disease, and I'm going to focus on the disease typhoid. And I spent my life working on typhoid. It's a disease of the impoverished, still very common in many parts of the world, and I'll talk a little bit about it because it's a human-specific disease. You can infect any other animal on the planet with Salmonella typhi, and it will not go into that. Even chimpanzees, it doesn't cause typhoid. It's really very, very well adapted, exquisitely adapted to the human being, human organism, and there's many diseases actually like that, and we've learned a little bit about why that's the case. But I also want to try to say, well, can you use science for impact? Can you do science, but also have impact in terms of changing policy or changing, if you like, the use of treatments and medicines in society, particularly in low and middle-income countries. So that'll be the sort of theme of what I'm trying to get across to you. So this is, for those of you who are not aficionados of typhoid, it could be any disease I'm talking about, but typhoid is my speciality, and it's really an enteric disease that has mutated and manifested itself, reinvented itself as a systemic pathogen. It is acquired by oral ingestion, gets into the gut, but it stealthily invades the intestine. It doesn't really grow in the intestine. I'll explain why in a second, but it actually stealthily goes in and causes a systemic infection where it resides mainly in the gallbladder, liver, and probably bone marrow, and this is the organism that causes it, Salmonella typhi here. And it really is involved in replication and persistence. It probably adapted to humans, thousands of years ago, where it normally causes a silent infection. Probably most people get infected with the organism but never get the disease. The disease is an accident, an immunological accident, really. And one of the key factors of the organism is the carrier state. You'll all have heard of typhoid Mary. And I've just recently been doing some studies in Chile where they eradicated typhoid from large parts of Chile in the 1980s. But grandmothers are now infecting their grandchildren this year with typhoid. And these are the carriers actually of that 40, 50 years later are still causing infections in Chile and in Santiago. It's a really incredible organism. The other interesting thing about it, which I won't dwell on, is it's almost impossible to isolate from the environment. It's very difficult to grow, very difficult to isolate from the environment. We don't really know why, and we're trying to find it. It may go into some quasi-dormant state that we don't know about, but it really has a remarkable life cycle. We tend to study this part in the laboratory, but it's really the whole life cycle that makes the organism successful. And when you look at the distribution of typhoid around the world, you get this sort of a map, which is based around the level of mortality and morbidity. And there's probably a vast underestimate of the impact of typhoid. It's underdiagnosed. And people that get typhoid are actually ill for quite a long time, and they fail to thrive. And it's one of those diseases that impairs the development of children in the third world. It really is one of the, it's got a lot more problems than just the simple acquisition of acute typhoid disease. It sort of hits the ability of people to thrive. And it's really a patchwork, because if you go into different parts of Africa, for example, you'll find typhoid is very common in one part of the area, and one part of the country it's not very common, and it's probably something to do with the persistence and carriage of the organism in terms of its dynamics in the human population. There's some other typhoid-like diseases. There's paratyphold, which is really associated with Asia, and there's also a disease called invasive non-typhal salmonella, which is a newly emerging disease that looks like typhoid in Africa, which I have a slide on towards the end of my talk. So what is the organism? Salmonella typhi, briefly, it's a bacteria. It's a member of the Salmonella enterica, but it's a monophyletic lineage. And if you look at the genetic map of Salmonella enterica and look at the genetic space occupied, Salmonella typhi occupies a very tiny part of that space. It's very constrained in terms of its genetic diversity. Salmonella paratyphi is over here. It's an independent introduction into the human population. It also produces this thing called the VI capsule, which you see here. This VI capsule has been, it's Achilles heel because it's been used to make vaccines against Salmonella typhi recently. And there's a toxin which was recently discovered by Jorge Galan and Stefania Spanner, who's in the audience, and showed that it probably plays an important part in the persistence of the organism. So we know a little bit about the organism. They have this carrier state. Probably entered the human population several thousand years ago, but the key to it's got a degraded genome, and a degraded genome is a signature of adaptation to a host. And we see this, we saw this first when we sequenced the genome in around 2000. We noticed and what it's really doing, it's throwing out a lot of the opportunities to cause pathogenesis in other organisms and specializing in infecting hosts in a particular way. It's very well adapted. The other thing, this is where actually it's never been published, but it's never been seen before. This is actually Salmonella typhi sat in the gallbladder of a human being. It was generated by Steve Baker, who worked in my laboratory. He's also a former Fleming Award winner, Steve, actually. I've got 2 Fleming Award winners who have been through my lab, and Steve was one of them. And we got this gallbladder. It caused me all sorts of problems when it arrived in the Sangha, because we didn't have ethics for it. So I had to sort of send it back to Nepal, then bring it back again. Then I said, you can't send it back to Nepal because it's not ethical. So I was in this sort of dynamic situation where I was unethical whatever I did. But fortunately, we managed to get through that one. And this is Salmonella typhi. It's actually, this is the VI antigen. It sits actually in the lumen of the gallbladder. It's not inside organism, it sits there, and this is how its sort of persistent state is in the gallbladder. It's very interesting to see and observe that. So in summary, the pathogenesis, this is what happens when you get a sort of a gastroenteritis type infection. You get a full-on assault, from salmonella. It replicates very high levels. If you get food poisoning, you will be shedding salmonella for several months afterwards and still be infectious. Whereas salmonella typhi, it's lost the ability through the pseudogene formation to really replicate in the intestine. It relies on replication in the gallbladder, and it sits in some form of macrophage, probably, as it moves through the body, and then is shed in bursts as sort of showers of organisms in terms of transmission. That's really a summary. And it's got VI antigen, which I'll show you. It has an anti-inflammatory effect on the mucosa and dampens down the mucosal response. So one of the problems I had for many years was I couldn't study Salmonella typhi in the laboratory. There was no animal model to study typhi. And this held the field back tremendously. It also prevented the development of vaccines against typhoid as well. And so what to do about it? Well, what you need in science sometimes is brave individuals or foolish individuals, maybe a combination. But this is a brave individual. So I got a phone call from Andy Pollard about 10 years ago and said, Doug, I want to set up a challenge model for Salmonella typhi in humans in Oxford. And I sort of checked, I thought, Andy, are you serious about this? You know, are you actually going to set up a human challenge model for this disease in Oxford? Because there had been challenge models in the 1950s. And he said yes, and so we wrote a grant together and we went for interview, and it was the most brutal interview I've ever been in. I don't know if any of you have been in fellowship interviews, and one of the external experts was accusing us of actually endangering Oxford, and we were going to cause an epidemic of typhoid in Oxford, and I was like this, ducking and diving, my God. And Andy's this calm pediatrician, he just sat there and sort of took it all on calmly. And as I walked out the door, and I said, if you don't fund this, you're going to set typhoid research back 50 years, and then walked out the door, and we thought we'd lost the grant. But actually, at the Wellcome's credit, they funded it. And I can tell you, this is not only shaping the way in which we register medicines for typhoid, it's going to change the way we register medicines for many, many other infectious diseases, and I'll explain why in a second. So off we went. Andy went off and developed this challenge model, and we got this strain from Mary Quayle. Mary Quayle was somebody who lived in the 50s. She was a carrier. The strain had been isolated. It had been put into a freezer in Baltimore, and we arranged for that, and it was used in human challenges in the 1950s. We arranged for the strain to come back, and we grew it up in our CL3. Del Pickard in my lab grew it up. We sent it for GMP preparation down at Porton Down through Steve Chatfield, and then the first volunteers were challenged. We thought we'd start at a very low dose. Remember, this is an organism that's been in the freezer for 50 years. And you can see the attack rate of 55% at 1,500 organisms. And remember, this is an organism that will not infect any other species on the planet, and it's exquisitely designed to infect you. The interesting thing is, even when you went up the attack with a higher dose, the attack rate didn't go to completion, that some people seem to be inherently resistant to typhoid challenge, at least in that context at that time. And you got the classical appearance of the organism in the stool, and I said it didn't really last very long in the stool, and then you got the appearance in some individuals in blood culture, and this is one of the volunteers. They got plenty of volunteers, mostly medical students, but they've now, I think probably over 500 to 1,000 people have taken part in these studies. And you get classical typhoid fever. We get the emergence of fever, and if they get diagnosed with typhoid, they get treated with antibiotics. If they don't get typhoid, they get treated at 14 days with antibiotics. And this was published. And there's been many studies now on sort of how typhoid manifests itself. These are PCR assays or culture assays for the organism. You get this classical typhoid disease, latent infection, PCR positive. See, the first thing we noticed was that at 24 hours, you got a PCR blip of the organism, and then later on, individuals went on to get typhoid or they didn't. And we'd never been able to study typhoid in this sort of detail and in this sort of way previously, and with a very controlled way in the population. And what we saw was this signature, which we're still looking at actually, which is in all volunteers, everybody who got challenged. So sometimes if you challenge them and they don't get the disease, well, did they see the organism? But everybody who got challenged, you see this signature of infection, but only some of those individuals then go on to cause disease, to get disease. And this is really and epithelium. We've studied at great length, I won't go into it today, but we studied at great length the sort of immunology of typhoid and use it for diagnosis development, looking at maybe markers for metabolism, etc. And it's a fantastic system for actually investigating the organism in terms of how it actually manifests as a disease. But really, in summary, what this is the first phase is really a luminal. It's a signature in the blood, but it's really as the organism passes through the epithelia. And the second phase is dominated by a macrophage signature. It's really a macrophage-mediated disease. A lot of signatures that you see in the volunteers are actually macrophage-type signatures. So we went on, actually, and Andy then did a phase 1, phase 2 study where he actually used a vaccine, which is a conjugate vaccine. Remember that we've been working on typhoid vaccines for all of my career, and we were never able to move it forward. We required a phase 3 study of £20 million to actually do the next phase. We were completely blocked in actually developing a typhoid vaccine. When this data came out, it was revolutionary in the sense that he was able to show that this Typhi conjugate vaccine was protective in these individuals. A nice paper in Lancet, and it was supported by the Wellcome were very brave in supporting it through a strategic award which I helped organize. And then Gates came in and developed, we moved this into the field, and then we carried out field studies in Nepal, Malawi, and Bangladesh. And I was at a meeting last week in Hanoi where they announced the first interim results of the VI vaccine study, and it was like one of these things that you get on TV where there's a competition say, and the winner is, and I was thinking, they were going to announce it, is it going to be 20% protection, is it 30% protection? I thought it would be around 50%, but actually, it was 80% protection. Early days yet, but this is going to be revolutionary in the way we can now use it politically to get politicians to realize that they can control typhoid in the field. This would not have happened without the human challenge study. We were able to bypass. We went to SAGE, and I'll explain exactly how we did it in a later, but it's also the way that cholera vaccines are now likely to be licensed. And since we set up the CHIM in Oxford, the Wellcome Trust are going to also set up CHIMs, or human challenge studies, for a whole variety of diseases, and they're going to move those challenge studies to endemic areas where the vaccines are needed. And this will change the regulatory route to all how future medicines are registered. And I think it will be used for antibodies, and I think it will also be used for antibiotics in the future. And also, you don't have to use animals. So it's actually relieving on the use of animals. So this is a really important point I want to make about impact and how you make impact. So where next? So what I was interested in doing was that can we reproduce the aspects of typhoid infection in an in vitro model that we can study in the laboratory? And so at Sanger, they were using this new approach, which was human-derived induced pluripotent stem cells, which they derived from the skin or blood biopsies of volunteers. Basically, you deprogram the cells into pluripotent stem cells, so you can then redifferentiate them into almost any type of tissue or organ. And this is something I was watching develop alongside me at Sanger for people using Yamamoto technologies. So we set a system whereby we could take these stem cells from people with rare diseases, we call them IPS, and also for people with common genetic variants, and make depositories of these cells. And we set up in the lab the ability to the same cell can move it in the direction of an organoid, an intestinal organoid, or we can move it into the direction of a macrophage. This was all set up by PhD students and people in the laboratory. We have it working beautifully now in the laboratory, and we can move them into dendritic cells as well, all from the same patient. And you can stock these and can reproducibly make these organisms from master cell banks. And what you can then do, this is sort of the protocol. They're quite intensive protocols to do this. And so once we've got the organisms, these might be macrophages here, we can do all sorts of assays on them, which the omic assays, and really get down to the nitty-gritty of what the responses are to the pathogen in these systems. I won't talk about the macrophage side. I'll just talk about the organoids quickly. These organoids are beautiful, actually. These are images they produce filling. These are enterocytes, mucous goblet cells, there's panas cells, and there's enterocrine cells, all displayed on these epithelia. And what we've set up in the laboratory, in the CL3 laboratory, is the ability to microinject these. This was done by Emily Elise, who's a clinical PhD, and Jessica Forbisher and Amy, and Chris is also in the audience. And we set up this beautiful system for microinjecting organisms into these organoids. Yeah. So you see that this is how we microinject them. It's not easy, but you come in with a needle. This is Emily doing this, and she can do about 300 in a morning. And this is just a stain that you use to show the organism going into the organoid and then off again. And then she goes off and does another one like this. It's quite a nice system. So you come in and do another organoid. So little mini intestines. And when you put the organism in, this is Salmonella in the lumen, and then this is an enterotoxigenic E. coli stain with phalloid, and you can do all sorts of, we're interested in doing rotavirus next and maybe other organisms. It works beautifully in the systems, and I know Nick and Chris are doing cholera in these systems. There's a poster actually on the cholera work if you want to see that tomorrow. So what have we done with this? Well, actually, this is the sort of thing you can do. This is a paper we just published in PNS where we discovered a new IL-22-mediated bacterial phagocytic capture system whereby IL-22 induces a barrier function activity in the epithelia that actually captures and destroys bacteria. You can see this killing, this is SL134, a Typhi, and this is after IL-22 treatment. We also found a patient who we had an IPS line who was a child who had early onset IBD, and they have a mutation, this IL-10R2 receptor is part of the IL-22 receptor. And that was why the patients were getting early onset IBD. And we've actually been able to treat this patient with stem cell therapy. So this technology, not by CRISPR repair, but this technology can be used, I think, in the future for repairing a lot of genetic and defects. What we were also able to do then is to define the IL-22 pathway actually in these organoids and identify lots of novel genes that were upregulated by IL-22. And here's the receptor. This is Dave Gooding, who I've had the privilege of working with, one of the best imaging technologists in the country. And this is the IL-22 receptor on the surface of these cells, beautifully stained by David. And this is the patient here who loses the ability to capture and kill the salmonella. The patient line does it, the wild type line does. But when you complement, we reintroduced a functional IL-22 gene back into the patient's stem cells, and we're able to restore the ability of those cells, showing that it was that mutation that was responsible for the phenotype. The clinicians thought it was an IL-10 mediated phenomenon in the patient, but it was also an IL-22 mediated phenomenon in that child. And we've been able to go on and do a lot of other similar studies with other mutations using other patients. And I think that's the sort of kind of experiment we'll be able to do in the future. We also identified, and we noticed in the IL-22 signature that we have this gene calgranin B, and one of the referees said, well, why don't you knock this gene out as well and see whether it has the same effect? And actually, it previously been implicated in capturing MTB in macrophages, and it turned out that this is the knockout, CRISPR knockout over here, that it loses the ability to mediate this IL-22 capture. So that then becomes another potential target for therapy of a molecule in the epithelia, and we're sort of building up a repertoire of information about how these molecules might work on the human epithelial rather than a model. We've recently infected them with Salmonella typhi. These are the strains that were used in the human challenge. And Emily has shown that they beautifully infect human intestinal organoids. So we now have an organoid system potentially for Salmonella typhi and Paratyphi. This is the quail Typhi challenge, and this is the Paratyphi challenge that Andy's been using. And we've got beautifully this is a Salmonella typhi with its VI capsule on the surface. And this is a Salmonella Paratyphi A, and we see this beautiful, I think it's a type 4 pili, which would be quite interesting to investigate. And we've seen a very beautiful signature associated with Typhi in these organoids. And we're now going to compare that signature with the signature we see in the volunteers at the early onset, the 12 to 24 hour time period, to see whether we can actually bring the two closer together to get more idea of how Salmonella and other bacteria are restricted at the mucosal surface. So this is all work that is ongoing in the lab at the moment. But for the last part of the talk, I wanted to move away from the lab out into the field. So the other thing I do is I spend a lot of time out looking at typhoid in the field. And I have a slide here which shows me, and this is recently in Madagascar where we have a field. So this is John Clements, we're sat on the veranda, and John is probably the most eminent vaccine epidemiologist in the world. He was formerly the director of the International Vaccine Institute. He's now head of the director of the ICDDRB in Bangladesh. He run the DOMI project, which Gates funded for $40 million, which categorized disease burdens all the way across Asia, and he showed VI vaccine would actually work, and he showed the impact of not only direct protection, but also herd immunity. This is a guy, Florian Matz, who I recently have recruited partly into Cambridge. And Florian spends his life in airplanes setting up field sites. And so I'll give you a little anecdote story, is that how many PIs in this room have applied for a grant, got overheads, and seen their overheads disappear into the mist and never seen them again? Put your hand up if you've ever been frustrated by that experience. Almost all of you. So Florian, this is a great story. He was in Madagascar. He got this £15 million Gates grant. And he made sure that the overheads went to Madagascar. And the Madagascar said, well, what do we do with overheads? And so what they did was they built a building where they did all the microbiology. They actually named the building after Florian. So I was thinking to myself that, how come we don't get things named after us? We could have the Nick Thompson unisex toilets named after us, something. Or we could have the Paul Kellam parking bay at Imperial College, but we don't even get that, do we? We just have to put up, but he gets a building named after him, but good luck to him. Anyway, on to the real story. So the reason I got involved in genomics was this reason. This is me actually as a PhD student here. And I look exactly the same, of course, as I did then. But I actually worked on this plasmid COLE1, and my PhD was actually mapping the genes on COLE1. This is reference number 3. I was very proud of that. I used to catalog the number of references that I had. And this was number 3, but this is 4.2 kilobase pairs long, and I never imagined in my life that when I was a PhD student we'd ever sequence that 4.2 kilobase. I thought, bloody hell, this is... I remember I was very lucky that when I went to a meeting in one of these meetings in the US, and I was in the back of a car as a PhD student with Beckwith and Gilbert in the front seats. They will never remember it, of course, but they were just taking us out for a drive. But I've gone from here to the idea that we can now sequence millions of base pairs and literally millions of genomes. I tell you now, as the young investigators, you would never imagine, 20 years on, 30 years on, 40 years on, whatever it is, that you could do this. And I'm very, very optimistic, actually, about where we are. I think that the young scientists of this generation are going to inherit the world in terms of, I think we can have enormous impact in the next 20 to 30, 40 years if we set about it and we protect them, that cohort, and get them working on the right problems in the right attitude. I'm still a great believer in the need for good, high-quality science to fight false science. This is really important. That's my sort of last philosophical interjection. But we set about, so I had no idea what typhoid looked like in the world in the present day. So we formed this consortium. We didn't get a grant for it. We just decided to form a consortium where we invited the community in to actually start to sequence Typhi genomes and get a snapshot of what typhoid looked like in the real world. And I did it through PhD students, actually. And these are Kat Halter's here today, and Kat was absolutely brilliant in this, with help from Julian and other people at Sanger. And she did some of the very first analysis of using sort of Illumina combined with other forms of sequencing and develop a lot of the tools. And these are other students, Sandra here, who's recently worked in Africa, Vanessa Wong, who was a clinical PhD student, and Zoe now doing this, who just curate these databases. We're now up to about 3,000 genomes, and we've covered many, many countries. We're going around the world now filling in the gaps. And when we first got this data, I was completely shocked by what I saw. And we've got hints of it when we did some work with Mark Ackman. We published a paper in Science which used haplotyping. But This is the family tree. I said that Typhi had only ever come into the human population once, and it's got a common ancestor, and it's every single Typhi in the world has originated from that common ancestor in the human population. It's really quite remarkable. And these are the older Typhi, and these are the sort of newer Typhi as you go around. But there's this spur that comes off around here, which is called H58. And if you look at the modern Typhi, almost all of the modern Typhi are H58. And what's happening is we're getting replacement, we get an extinction of all old forms of typhoid and replacement with a new form of typhoid that we've created in the last 20 years. And when you look around here, these little slashes here are actually antibiotic resistance signatures, and the yellow and red are the more antibiotic resistant. You can see that what H58, it's an AMR form of typhoid that actually is now replacing all of the typhoid. The other interesting thing is typhoid used to be very rare in Africa. People said that typhoid didn't exist in Africa, and there was a guy, Sam Kariuki, who I was working with in Kenya, and we spotted typhoid and H58 actually arriving in Kenya in around the beginning of this century. And I said, well, I think that this is going to spread into the rest of Africa. And I went down to the Malawi unit where the Wellcome Trust said, I think you're going to have a typhoid epidemic going to come your way. And they said, no, we never get typhoid here. It's not going to happen. And actually, this is what happened. This typhoid arrived with a vengeance. It's still there. And this is H58. This is what typhoid looked like before H58 arrived. And this has now gone on into Zimbabwe. It's in Zimbabwe, and there's a global emergency. And because we've got this typhoid vaccine, conjugate vaccine, Gavi are actually implementing now mass vaccination. Zimbabwe is called in the vaccine and trying to control this outbreak of H58. So not only do you get an AMR, but you also get a redistribution of the disease around the world. And it's rather like we're seeing, I sat at the Sanger, it could have been C. difficile, it could be MRSA, it could have been a Shigella, it could have been a cholera outbreak we've been mapping. And we're seeing the reorganization and the redistribution through human pressure of infectious agents. And Reno talked yesterday about the effect on the microbiota as well. And this is not just a phenomenon of typhoid, it's a phenomenon more broadly that we're seeing across the world. And this is a study we've recently been doing in Kenya with Sam Kariuki, and this is one site in Kenya, and really just to focus on this part here. These are the haplotypes that are causing disease in one settlement in Kenya. And there are at least 3 different, what we call H58 lineages here, which Zoe's been mapping, and Sam. And they're all different waves of typhoid that have come into the settlement, established themselves, and are still there. And they've all got different genetic signatures and different types of resistance. One's coming from Uganda, we know one's. And what we can now do is we can follow the movement of these organisms across the globe and decide where we're going to intervene in the infections. And also, these are all AMR. And with the beauty of whole genome analysis, you know, you can get in the minutiae in all sorts of ways. And what we're trying to do now is we've mapped a lot of these infections. You'll see it better on the next slide. This is just a geographical area. We got an NIH grant to do this, and it was funded for 3 or 4 years. And these are lineages. And we map them against public toilet, water points, sewage. And then we come up with this sort of heat map, which these are the water points are blue, but you'll see the Typhi dotted in a different Typhi. There's a yellow, orange rather, and red. And you start to see clusters of the organism. What we have seen, particularly in the Congo we've seen, is where we can now map outbreaks. So we'll be able to go in, look at find out in more careful analysis of how the organism is moving about. And we're now going to try to work with governments to go in, and this is the way that all epidemiology will be done in the future. They haven't realized it, the vaccine people haven't realized it yet, but this will be the way that we intervene in diseases in these settings in the future, and it'll save us a lot of money. We won't have to bring vaccines in across the country. We'll probably target vaccines into certain areas. This is a really exciting time, I think, for people in the field. It's not over yet. I was rang up again from Pakistan about 2 years ago, where they said they've got an incredibly resistant organism which is spreading in Pakistan. And would we have a look at the isolates from that outbreak? And we did, and we got the isolates very quickly across and sequenced the isolates. And it turns out that the isolates in Pakistan are a new form of H58, which is this little tree here. It is a real outbreak. There's very few SNPs. We sequenced around 200 isolates. It's now gone, it started off in Hyderabad in Pakistan, but it's spread across Pakistan. And again, there's an emergency. We use this publicity to get investment from the Pakistan government to put political pressure on them to now intervene and vaccinate against this XDR form of typhoid. Interestingly, Vanessa Wong, who I mentioned, is a clinical student. She was in Addenbrookes Hospital and somebody presented with typhoid, and it was an XDR typhoid that she had studied from Pakistan. And I thought it was quite ironic. There was a clinician, and she rushed up to me and said, dude, we've got an XDR patient, a typhoid patient down in Addenbrookes. And I said, all right, go write the paper, Vanessa, you can get paper out of it. But she'd also saved the patient. And this is another so this is not just a one-off. We've noticed in the Congo also that there's another form of XDR type typhoid, which is not a H58, it's another form. So these things keep popping up periodically and are real problems. And this is almost my last slide, and this is Elizabeth Clem, and it turns out that this is the most resistant organ I've ever seen. It's got an integral on the chromosome, it's got lots of SNPs associated with fluoroquinolone resistant, it's got extended XDR resistance to various antibiotics. But also it's acquired a plasmid, which we've actually a very promiscuous plasmid, which is all over the world actually, and it's an ink-Y plasmid. It reminds me of Stan's philosophy that antibiotic resistance itself is infectious. So I went back to and I did this for Nigel Brown actually, I think he's in the audience, that actually, interesting, this is from my PhD, where how we used to map the integration sites of transposons, and this is TN1, which encodes beta-lactamase, and this is TN501, which encodes mercury resistance, and they're both actually in this XDR Typhi. So I thought, connection right between my PhD and now me working on these XDR organisms in Pakistan, actually quite remarkable. Last slide really is this is not all over yet. There's been a re-emergence of a new form of typhoid in Africa discovered by Sam Carrick. He's one of the most remarkable people I've ever met in my life. He's one of the true mentors and leaders in Africa. You know, he discovered this, and nobody in the West believed that there was this new form of typhoid that was going from human to human non-typhoid soil. And I met him in Malawi and said, Sam, let's have a look at it, and it turned out to be absolutely right. We mapped the spread of this strain. These are the ST313s. They were driven by the HIV epidemic, and they moved right across Africa, and they're still moving across Africa. And there was a 50% mortality rate. I went into a hospital in Blantyre, Malawi, and there were people on the floor dying of this disease, and it's incredibly nasty type of infection in malnourished children. So in the end, then, so the impact of this AMR data was quite significant, because when I was invited to WHO to the stage where they sort of approved the next stage of vaccines into deployment, and they actually asked us to present on the typhoid case on the impact of AMR. And so it was the first time ever I'd seen AMR impacting on the introduction of a vaccine. And WHO took it serious, and it helped get the vaccine pre-qualified and licensed. And so people are now bringing a lot of this data together to sort of measure impact. And I encourage people to be positive about what you do and trying to look for ways in which you can actually have impact in these settings. I'm going to finish now, so I think I've finished on time, which I think is good. I always try to finish on time. But I've got this slide, which is acknowledgement of my current team. And I wanted to pay special tribute to Sally, Forrest, and Josephine, who've helped me set up it's flipped too quick, it doesn't matter. But what I've did is, you can't name everybody who has ever worked in your lab. But what I decided to do was to send this present me with the prize. In fact, a lot of you in the audience will be on this, and you might recognize people who have actually worked in my laboratory. There's 2 Fleming Prize winners. There's a Vice Chancellor of an Australian university. There's a Director of the Wellcome Trust. There's many professors of microbiology in and around the world, and just see if you can spot any of these individuals. You can see Matt Palin there, for example. But just look at that while I finish. Okay, thank you very much.